Hi, this is Miss Litton, and this is my noisy 7th period review. They are motivated. Say hi. Hi. It's terrifying. So this is our last official review of 2018. Because next Tuesday, your bangs look super cute. But next Tuesday, she's like, why are you drawing attention? Next Tuesday, we're sorry, we're tired. I think I say that all the time, so I don't think it works anymore. Next Tuesday, I'm just open for help. Monday, I'm open for help for Honors Day of Priority, and then Tuesday, I'm open for help for you. If you have last-minute questions that you want to just come in and ask me about, okay? All right. So, Unit 3, top 5 topics. So the first one is cell cycle, steps, and regulation. So when I think cell cycle, I think the bulk of the cell cycle is in what? Interphase. And interphase is broken up into?
3 stages.
And the first one being? G1. Second one? G2. Good. So just review a picture of the cell cycle and what happens at each stage. And its regulation. Do you remember how we talked about there are checkpoints it has to go through, just like you go through security in an airport, right? And what else? What was another regulating? You needed to have what? Cyclins. Yeah, cyclins like your ticket. M cyclin and S cyclin. M cyclin to move into mitosis, S cyclin to move into synthesis of DNA. Do you want these? Is that what you're asking for?
No, it's
Okay. Mitosis versus meiosis. Step, significance, mistakes, and cancer. So much there. Okay, on your mitosis, meiosis, be prepared to answer a question. If I say your N number is 2, diagram metaphase, or metaphase 1, or metaphase 2. Could you do that?
Yeah.
Do you want to do that together, or are you cool with that?
Let's try.
Oh, never pause. Okay, so let's do this. N equals 4. What does that tell us? If I tell you N equals 4, where would you find just N equals 4? In a what? Haploid. So this could be a ginormous gamete, right? But that means your 2N number would be what? 8. So this is a cell with 8 chromosomes. If I tell you the 2N number is 8, then you would know the N number is 4. You totally have that. Okay? Then could you draw, let's just pick all the different, mitosis. So all the metaphases that you could possibly have. So you could have mitosis, and then you could have meiosis, metaphase 1, and you could have metaphase 2. Do you agree with that?
Yeah.
Okay. Could you, in your mind, imagine what those cells would look like?
Yes.
Is that what you imagine for mitosis? Okay. And what did you imagine for metaphase 1? Is that what you imagined? And then what did you imagine here?
2 cells.
Do you agree with that? Okay, so it's terrible. That's an easy way for me to check that you know how to do those stages by just asking for one stage. I could ask for, the stages that are really easy for me to check on are like metaphase, anaphase, prophase, all the phases. I just have to name a stage and then give you a number, and then you should be able to draw it accurately. And that will tell me if you understand it. Okay? So on mitosis versus meiosis, why would you do mitosis?
Just regulate.
Growth and repair is why you do mitosis, right? Growth and repair, or asexual reproduction, you might say, right? Whereas when you're doing meiosis, that increases what? Variety. Yeah. How does it increase, or where can I point to as this will increase variety? What's one of them? Tell me one. Crossing over. What is crossing over? Exchange of What? Alleles. Okay, be more specific. Exchange of, I'm hearing it. Non-sister chromatids, homologous pairs, right? Exchange of chromatids, right? In a homologous pair, but non-sister, right? Because this is a sister chromatid and this is a sister chromatid, and if they exchange, it doesn't do any variety, right? Non-sister. Okay, and when does that occur? Prophase 1. Prophase 1. Okay? That's one way meiosis increases variety is through crossing over. What's the second way? Independent assortment. When does that occur? Metaphase 1. Metaphase 1, right? Okay? And what's the third way? Fertilization, because you know what sperm's going to hook up with what egg in your diploid zygote. So those are the three ways we get variation. Why is variation so significant? What hinges on variation? Thank you, natural selection. So we needed to have a source of that variation, right? That's why we said Darwin. Hi, how are you, Lindsay? Are you coming to visit a friend? Oh. Okay, fine. All right. So you needed a source of variation in order to have something for natural selection if you're going to have that work, right, as driving evolution. Okay, and I don't know why it does that. Okay, thank you. All right, steps, significance, mistakes in cancer. All right, so let's talk about cancer. What are the two genes we need to know about for cancer? Tumor suppressor, yeah. Okay, so proto-oncogenes are normal functioning genes that happen all the time, right? Doing their job every day. Promoting what? Cell cycle. Tumor suppressor genes, normal job every day. They are promoting what? Apoptosis. Stopping, checking, and if it doesn't work, then death, right? Those are tumor suppressor genes. So you have to have it go wrong in both of them really to get cancer, right? Because you have to have uncontrollable growth, which means that a proto-oncogene must have mutated into a, which is promoting the cell cycle, maybe generating some sort of cyclin that promotes the cell cycle, and you're not able to take care of it with a tumor suppressor gene. What was our example of a proto-oncogene? RAS, and it promotes the cell cycle. Remember, it's the RAS gene coding for the RAS protein. The RAS protein is a receptor on the cell membrane that binds with a growth factor, stimulating growth through cell division, right? And normally that has to be stimulated, that receptor has to be stimulated in order to promote the cell cycle, but it is getting, without any kind of hormone, it's getting triggered to promote the cell cycle. Yes?
And it can bypass the cyclins, right? The checkpoints?
Or it can turn the cyclins on that it shouldn't.
Right.
Okay? Tumor suppressor genes have the cell cycle stop, so it can be fixed. Remember, we want to check right after we replicate the DNA and make sure we replicated the DNA correctly, and you also have a checkpoint right after you've done mitosis to make sure you did mitosis correctly. And once you stop, repair, but if you can't repair, then remember we said promote cell death. Okay. What was our example of that one? P53 gene. Good. Okay. On cancer, we have characteristics of cancer cells. Can we think some of those characteristics? Loss of anchorage dependence, loss of contact inhibition, multi-nucleated, right? Abnormal nuclei, abnormal cell surface, right? Metastasize can spread to other places. Angiogenesis. I can't hear what you're saying back there. Chromosomal, variation. Angiogenesis, blood vessels start going towards you. Those are all things that are irregular about cancer cells. Okay, good. Can we let that go? Are you good with that? Okay, so let's move on to inheritance patterns. So we'll start with Mendel. Mendel has 3 laws. What are those 3 laws? or principles. Law of dominance, like tall being dominant over the short allele, right? T over little t, right? Does that always hold true?
No.
No, dominance does not. What is an exception to that? Okay, let's name several of them. Sex-linked inheritance. Because normally, in order to express a recessive trait, you need 2 recessive alleles to express a recessive trait. But in males, since you only have 1 X chromosome, it doesn't matter if it's dominant or recessive, you'll still express that trait because you only have 1 shot at that trait. Okay, that's one exception to the law of dominance. Can you name another exception? Codominance. And one you're always going to use for that is what? Blood type. And the codominance is seen in that you have big I A and big I B, which means you have surface antigens of A and B, both on the same red blood cell. Okay, and that's also an example. Blood type is also an example of what? Multiple alleles, because we don't just have big I, little I, we have big I A, big I B, and little I, which increases not only the potential genotypes we can have, but also the potential phenotypes. Good. What else? Incomplete dominance. Incomplete dominance looks like blending to our eyes, but it's not. You just have a little bit of each trait expressed. What was our example of incomplete dominance? Yeah, red flower crossed with a white flower, and the incomplete dominance part is when we get pink. Curly hair and straight hair, and we get wavy hair. Okay, what's another example? Exception, yes?
Incomplete penetrance?
Penetrance would be, yeah, I guess you could say that too, because you may not totally express that trait, even though you have the genes for it. I'm looking for another one that's kind of obvious.
Polygenic?
Yes, that's exactly what I was looking for. When a trait is coded for by many different pairs of alleles, polygenic traits. It's not straight up dominant or recessive. And usually you get a bell-shaped curve with that, right? With lots of different phenotypes. Okay, did we hit all the laws of dominance? Okay, what would be the next law? Law of segregation. And what is that referring to?
Each trait has 2 alleles.
Yes, each trait has 2 alleles, but what does the law of segregation refer? That they will be what from each other? Separated. When does that actually happen? Yeah, anaphase 1 and anaphase 2. And failure to do that is called good. Are there exceptions to that rule? Not in normal development. There are issues with that when that fails, right? Down syndrome would be an example of that, right? You have an extra copy of the GART gene. Okay, law of segregation. What's the next one? Law of independent assortment. Are you raising your hand, sweetheart?
Oh, no, sorry.
Okay, so law of independent assortment says what? Who assorts differently? Each homologous pair assorts independently of all the other homologous pairs. But see if you can use another word other than the name. You know what I mean? Don't use the definition in your definition, or don't use the term in your definition. Don't say, the law of independent assortment is chromosomes will assort independently. That doesn't tell me that you know.
Line up.
Arranged, good word. Each homologous pair can be arranged differently from the other homologous pairs, which then increases variety, right? And the exception to that rule is linked genes. And what breaks up linked genes? Crossing over. Okay? And independent assortment, that's when we looked at something like if we had big T, big R, little r, you could get big T, big R, big T, little r, little t, big R, little t, little r, right? That's when we did FOIL. Okay. All right. So let's see what we did. Simple, beyond multiple alleles, sex-linked. We did that. Polygenic, incomplete. Oh, what's pleiotropy?
Well, the mutant gene affects one.
Multiple phenotypes for one pair of alleles?
Yeah, like sickle
Okay.
cell disease.
Like sickle cell disease, exactly. Okay, good. All right, questions, concerns about that? Yes?
Can you go over epispaces?
Yes, what a great one to say. Okay, so for instance, if you have an epistatic gene, let's say for color, okay, for birds. Okay, big C anything gives you color. Big R anything, you will be red. Little r, you will be black. Okay, so you could have this, and what would that hen be? Red. Okay, and if you had this, what would that hen be? No color. Probably it's going to be some sort of like albino because no pigment is laid down. So it doesn't matter if you're big R, or little r, or big R. You have to have a big C in order to express it. That's giving you the how-to, and this is just saying in which way you will express that, red or black. Okay, so the C is the epistatic gene. It stands upon the other gene. Okay. All right, what else on that one? All right, so let's move on. DNA history. What are some classic DNA experiments we know about? Griffith. Was there anybody before Griffith? That was our first one, right? And so Griffith, what happened there? There were 2 strains of bacteria, right? And one was lethal and one wasn't. But then he mixed a non-lethal one with a dead lethal one, and yet he still got transformation, right? And so the R strain became a smooth strain, even though the smooth strain had been heat killed. So that meant there must be something that the one bacterium is getting from the other.
Like a
Transformative substance, and what was that? Okay, that was Griffith. Who came next?
APBio.
Cloud and McCarty, like the law firm. And what did they say? That transformative something was what? DNA. Then what was the next one? Hershey and Chase. Right, they chased it down. Do you remember what they used with that? Bacteriophage on bacteria, right? And they labeled, they didn't know the structure of a bacteriophage like you do today. They just knew that it had part protein, part nucleic acid. So they labeled the nucleic acid with an isotope of P, because base sugar phosphate, right? P35. And then they labeled the protein with S32. Yeah. And that's because cysteine, S35 and P32. S35, P32. They labeled that sulfur is in the amino acid cysteine. Okay? And they used, really, important, expensive equipment like blenders, but they also used centrifuges. And they identified that the transformative something that was changing the bacterial traits was actually DNA, because they found the what?
DNA.
The P32 isotope inside. Okay, what was another one? Charga. And he would say what? However many adenines you have the same amount of, and however many guanines, same number of cytosines. He didn't know what that meant, but he knew that that's how it was. But it could vary among species, but at least the A's and T's were the same and the G's and C's were the same. Then who else? Rosalind Franklin. And she sent what through DNA? And she says, hey, it's helical in repeating patterns, and it's a uniform size. Then who put it all together? Watson and Crick, double helix. Okay, are you okay in that little DNA history? Okay. So then we want to know the structure of DNA, right? I would be able to do a simple diagram of DNA, just diagram it out. Okay? So we know that the rungs of the ladder are what? No, rungs. Like what you would climb, nitrogenous bases held together by, and between adenine and thymine, you have 2. And between guanine and cytosine, you have 3. That's why you tend to have mistakes in DNA, like through sun radiation, they call them thymine dimers, where one thymine will hook up with another thymine instead of the adenine across the street from it, right? We know 3 to 5, 5 to 3, right? We know the sides of the ladder are alternating what? Sugars and phosphates. Can you believe how much you know? Just like, sugars and phosphates, guys, it's easy. Why am I even here? Yeah, everybody knows that.
Of course.
Alternating phosphates and sugars, right? And we know about DNA replication. Let us walk through DNA replication, shall we?
Okay, let's do it.
Okay, ready? DNA unwinds. And you know what? There's one that I told you there. Topoisomerase. And what? Unzips. helicase, and we have a leading, SSB. We have a leading and a lagging strand, right? The leading runs from 3 to 5, the lagging runs from 5 to 3. Either way, the first thing we're going to need to do is put down some primers. One time, right? It's primase who lays down primers. Primer, and then on the lagging strand, primer, primer, primer. Multiple times because we have to keep waiting for it to unwind so we can work backwards on that section, right? And then once we have the primer down, we can use DNA polymerase 3, and he's going to work off of the primer, and where there's an A, it's going to bring in a nucleotide that has a T, and where there's a G, C, T, and he goes boom, 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 boom. On the leading strand, he just zips right through, and then boom, 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 right? And now we have primer, long DNA on the leading strand, and on the lagging strand, we have several primers and several sections of DNA. What do we call those sections of DNA? Okazaki fragments, and they're about how long? 1,000 base pairs long, and then the primer is about 10 base pairs long. Did I teach you that?
Yeah.
Okay, so now what do we need to do? We need to get rid of the primer, 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 primer. Who does that? DNA polymerase 1. He's the one to say that. Now, it's no problem hooking together the phosphate sugar, no problem hooking together nitrogenous bases through hydrogen bonds. The difficult part is when you pull it out and replace the RNA nucleotides with DNA nucleotides. When DNA polymerase does that, you've got to seal the phosphate sugar backbone because you don't just have like DNA polymerase 3 who was just doing it as he went around long, right? That's why you have to use ligase. And then these will twist up and form sister chromatids held together by a centromere that separate during anaphase. Perfect. DNA replication. Now we need to talk about transcription and translation. Okay, here we go. DNA unwinds and unzips. Helicase. We're going to take our lagging strand and put it away, and we're going to use what? Yeah, RNA polymerase binds to the promoter unless the pressure on the operator. Exactly. Or it has a hard time, RNA polymerase binds to the promoter if it has help from a transcription factor. Right, okay. Control of gene expression. I love it. All right. So RNA polymerase binds to the promoter. Boom, boom, boom. Make your mRNA. Peel it off. Go get your lagging strand. Zip it up. Put it away. Here's the RNA. Put on a cap of 7-methylguanosine and a tail, 100 to 200 adenines, and cut out the introns before it leaves the nucleus. Now this cutting out, is it the same way every time? I can cut multiple different patterns, right? Yes. So one gene, multiple proteins, right? And the stuff I cut out, this RNA, they could become small interfering RNAs who like go get into trouble later, right? Okay, cut out the introns before it leaves the nucleus. I'm leaving the nucleus. Now I'm going to take my RNA and I'm going to go get on a what? Small ribosomal subunit, and then a large. And every 3 bases on the mRNA is called a codon, which is complementary to a anticodon. On the tRNA, T stands for transfer. It's transferring an amino acid. The first tRNA lands in what spot on the ribosome? Just the first one, P spot. The next tRNA lands on the A spot. tRNA1 dumps its Pinot Grigio. I don't know where I got that. I'm not even sure. I'm tired. Okay. Drops its amino acid. Oh, I know why, because I was in the P spot. Okay, I got that. All right. I knew I was like, why don't I say that? Drops its amino acid from the P spot onto the one that's in the A spot, but that happens by the ribosome shifting and putting the one that's in the A spot shifts back into the P spot, right? And then that leaves room for the next tRNA to land in the A spot, and the ribosome shifts. And when you shift out of the P spot, where do you go to? E spot for exit. And this will go, 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 go until you have a stop codon. How many stop codons are there? 3. But our start codon is always AUG, A-U-G, and that's the initiation codon. Always goes from a diamond. What? Okay, wait 1 second, I have a question.
So P is
I can't hear, shh, go.
So P is for all the amino acids that they have, right?
The longest one, the longest chain will be in the P spot. I always think P like protein. It's really a different name, but that works for me. And then the A spot is where you have that single tRNA that's there. And then as the ribosome shifts, it moves the long chain of amino acids that was on the tRNA that was in the P spot onto the tRNA that was on the A spot. That's all, which moves into simultaneously into the, just a minute, moves simultaneously into the P spot as it goes along. Does that make sense to you? Okay, you're next.
So the order of letters, is it PAP or AP?
It's actually EPA, if you want to think about it that way. EPA, Environmental Protection Agency. Oh, we don't have one anymore. EPA.
Why isn't it like
You can think about it that way too. That's just how I'm.
Okay.
Yeah, that's how I always think about it. Go.
Where is cDNA made in this process?
Okay, cDNA. I know we think of like complementary DNA, but that is that all DNA is complementary. But when you refer to cDNA, that's when you're trying to discover what is the DNA that coded for a certain protein that you want to use mRNA and reverse transcriptase to get the DNA that you can then work your way backwards to find out.
But
So,
where does that happen? This, like,
it's probably going to happen in a lab or
No,
something.
I mean,
Like
like in the. Oh, right.
It's in a lab because you're working that. Okay. All right. So now, okay. So it's going to go to we have stopped. Okay, we did all that. That's good. Transcription translation. I think that's good. Do you want to look at the essays referring to that? Yes, I do. Look at all of them. Yeah, we're probably not going to have a chance to go through them, but let's just hit the main points here, okay? All new cells come from previously existing. What says that? Cell theory. The process of cell division. Oh, by the way, division of the nucleus is called what? Mitosis or meiosis. Division of the cytoplasm and everything else is called cytokinesis. Good. We talked about mitosis and meiosis. Why am I being a pointer? I'm a pin. I'm clear. Okay. We did that. I'm reading it. Are you reading it? We did that. Autosomal linkage, when you're on the same chromosome, they'll travel together. We did that. Did that. Epistasis, pleiotrophy. Did that. Okay. We did all of those. Is there any on there that we didn't talk about?
No.
Okay. Look, there's more. Did we talk about Hershey and Chase?
Yes.
Griffith? Michelson and Stahl? No. Michelson and Stahl, they're the ones, Watson and Crick, they came up with semi-conservative replication where each DNA strand serves as a template for the new strand. Michelson and Stahl just proved it using isotopes. You don't have to know that experiment anymore. They say you don't, who knows? Describe the biochemical composition, structure, replication of DNA. Did we do that? Yeah. Enzymes, protein synthesis, transcription, translation, we did that. A yeast cell in the early portion of interphase of meiotic division has 24 FGs of DNA. If the yeast completes meiotic division to form 4 haploid cells, how much DNA would you expect in each haploid cell? Yes.
So for a question like number 6, would you draw a diagram?
Wait a minute. I can go back to that in a minute, except I just asked about this question 8.
Okay,
Yes.
12,
right? Because it would be what? Not diploid, but haploid. Okay, now, go ahead. Question 6.
So for a question like number 6, would you draw the actual diagram of DNA and this?
On the AP exam, they do not expect you to draw anything.
Oh, okay.
Yeah. Now, it doesn't mean I wouldn't ask you to draw something, but yeah. But that's because, yes. Any questions on 9? Tall, dwarf, round, wrinkled. If a big T, big R, little r plant is crossed with this plant, what fraction of the offspring will be tall and wrinkled? Do you remember the product law? Okay. What offspring will be tall and wrinkled? Can you give me the genotype of tall? Big T, anything, and wrinkled?
Little
Two little R's.
Oh,
Do you agree with that?
Yes.
Do you see the question I'm reading? Tall and wrinkled?
yeah.
Okay. So what percentage, if you do that cross, can you see that put it in your head?
Yeah.
What percentage will be tall?
75%.
Give me a fraction here.
3/4.
3/4. And then if you look at this one, big R, little r, crossed with little r. Be careful. Don't assume you know the genome. You know what I mean? Make sure you look at it because a lot of you might think it was a heterozygote again. So what are the chances here?
1/2.
Yeah, 50%, 1/2. 1 times 3 is 3. 2 times 4 is
8.
That was terrifying. Yes, 3/8. Did you get that?
Yeah.
Okay.
Oh, there's more.
Look, there's more. In fruit flies, a gray boy. Supposed to say a gray body. He's a gray boy. He's very ill. He looks gray. A gray body is dominant to a black body. So what I would always do in that situation is I would go, big G anything is what? Gray. And there's 2 ways to do that. And little g is black. And normal wings are dominant to vestigial. Flies heterozygous for both gray bodies and normal wings were crossed with flies that had black bodies and vestigial wings. The following results. Okay, so flies heterozygous for gray bodies would be big G, little g, right? Are you okay with me? And normal wings, which would be big N, little n, yes? Is crossed with flies that had black bodies. What would black bodies be? What? Little g, and vestigial wings, which would be what? Are you okay with that? Okay, you know every single time this one's going to give what? A what? A little g and a little n. Do you agree with me on that? Every single time they're going to give a little g and a little n. If you had the law of independent assortment, you would expect big G, big N, big G, what? Little n, little g, big N, and? Right. So what would we expect then? Equal, even, Steven, right? The 4 different phenotypes. Do you agree with that? Is that how it turned out in their data? No. There was predominantly gray bodies and normal wings and black body and vestigial wings. So that's when you would start to, in your head, go, oh my gosh, these genes must be what? Linked. Because they did not exhibit independent assortment. And the only ones that didn't show that would be this, and this would probably be due to what? Crossing over. Follow? Okay. Below are the amino acid sequences for 6 species of for critical parts. Oh, FOXP2 gene. That sounds like something with development, doesn't it? You're like, I don't care. Each letter represents a single amino acid. The data can be used to determine the relatedness of 6 species, assuming that mutation rates are equal for all species over time based only on these data. Which letters, B, C, D, or F? So you only have to look at B, C, D, and F. Represents a species least related to the other species. So what are you going to look for there? What would you look for? differences, right? A, V. Okay? T, 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 E, T, I. Is everything else there? PK, 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 SSD. Uh-oh. Here's one that's different. TS, that all the, oh, there's a difference. All right, so which one is the least related? D, because it has the most changes. Do you see how that wasn't a difficult? It looks when you read it all and you're like, oh, look at all those letters, that's hard. You're going to get questions like that on your AP exam and you're going to go, I can do this. You're not going to look at any question and say, I can't do it. You're just going to be real methodical and go, I can do this. Yes?
Why is it D?
Why is it D? Can anybody put that in words? Yes.
It has two differences.
It has two differences. The other one, this one has one difference and that one has one difference. Right? These two are exactly the same. Do you agree with that? Okay. And in E. coli, the DNA is 24% adenine. Based on this, what percentage is guanine? Well, adenine binds with thymine, right? So 24 and 24, what does that equal? 40, what? 8. So what's left?
52.
52. If we're going to share that between guanine and cytosine, that has to be what?
Divided by 2.
So it has to be what?
26.
26. Thus endeth the lesson. Any questions? Good job. Do we have to take the final? Yes. But what do you get to do? You get to take it with a friend. Yay.
Why not
The
the
whole thing with Grace.
Why not the whole final with the friend?
Have a good evening. Maybe have a piece of toast. I love you. Make good choices.